Looking at cellular respiration, this is like, I know this test is a lot of material, but my hope is as I go over everything today, you'll kind of see that it's a lot more of a process to understand, and the things you actually have to memorize maybe aren't quite as much as you might think. This slide covers most of it. So remember, the purpose of cellular respiration is how we make energy in the cell. The idea is we take glucose, and we're taking glucose and we're trying to make ATP. ATP, those are like little tiny batteries, if you will, that our cells will use for energy. To make that ATP, we do an oxidation reaction. We oxidize our glucose. I told you about oil rig. Oxidation is loss, so you're losing electrons. So when we oxidize glucose, we're stripping electrons away from glucose, and we're then going to reduction is gain, we're going to add those electrons to this molecule called NAD+. And when you add those electrons at NAD+, and you reduce NAD+ by oxidizing glucose, you then create something called NADH. NADH, they call it an electron shuttle. Basically, it represents stored energy. We're taking the energy from glucose, giving it to like a middleman that is NADH, and then we're ultimately going to use that NADH in the electron transport chain to create that ATP. That's really kind of the story of cellular respiration. Glycolysis is a series of 10 reactions. You don't have to memorize those 10 reactions, but do know that like reactions 1 through 5, those were, we called it the energy investment phase. That's where you put in two molecules of ATP, you invested that. And then reactions 6 through 10, the energy payoff phase. You then get out 4 ATP. But because you had to invest 2 ATP, you only net 2 ATP in glycolysis. And how we get that ATP out is something called substrate level phosphorylation. The idea of substrate level phosphorylation is basically you had, if I draw like an enzyme, and that's a really bad enzyme, but like you had one of these like intermediates of glucose we attach a phosphate to. And then you have, in this enzyme, you also will have the ADP. The enzyme will catalyze the transferring of the phosphate from your substrate to ADP, making ATP. That's what they mean by substrate level phosphorylation. Contrast that with oxidative phosphorylation. Oxidative is referring to the oxidation reaction. That's where we then oxidize these electron carriers. We remove the energy from those electrons from those carriers through the electron transport chain, use that released energy to make a lot more ATP, 26 to 28 ATP. Okay? The product that you get out of glycolysis is 2-pyruvate. When you think of glycolysis, I told you to think of 2s. You get out 2-pyruvate, 2 ATP, 2 waters, 2 H2Os, and 2 NADHs. So 2, 2, 2, 2, 2. You just remember 2, that's really what you get out. Then, and the location of this is in the cytosol. That's the cytoplasm. It's outside the mitochondria. Now, if you have oxygen, so assuming oxygen is present, we then go through the rest of cellular respiration, because you need oxygen at the very ending part of the electron transport chain. So the cell's not going to waste its time doing pyruvate oxidation and the citric acid cycle if it doesn't have oxygen present. Okay? So if we have oxygen, then the pyruvate is going to go into the mitochondria, and the pyruvate gets into the mitochondria by active transport. I don't have a test question on that exactly, and I doubt the AP exam would ask you about it, but technically we have to use active transport to get the pyruvate into the mitochondria. We then do this one-step reaction called pyruvate oxidation. We talked about oxidation where you're removing electrons, so we're further removing electrons from that pyruvate, and we're going to turn pyruvate into acetyl-CoA. One of the things you have to be able to do is count the carbons. meaning like glucose is a 6-carbon molecule, pyruvate is a 3-carbon molecule. That's why we make 2 pyruvates, right? Because you're taking a 6-carbon and making a 3-carbon sugar. That's why there's 2 pyruvates. So we take this 3-carbon pyruvate and we're going to turn into a 2-carbon acetyl-CoA molecule. And in the process, when you do that, you end up losing carbon dioxide. You end up losing 2 carbon dioxides. And I'll show you that reaction in another slide. Then we take that acetyl-CoA and we put it into the citric acid cycle. In the citric acid cycle, you're gonna, when you do that, it's an oxidation reaction. We will also make 2 more of our NADHs, okay? Because we removed electrons from the pyruvate and we attach them to NAD plus, making NADH. Then we go through the citric acid cycle. This is also called the Krebs cycle. Krebs is the scientist that discovered the cycle. And we're gonna send the acetyl-CoA through the citric acid cycle. There's a lot of reactions there. Unless you take biochemistry in college and stuff, you won't have to memorize the actual reactions of glycolysis and the citric acid cycle. Just focus on locations, inputs, and outputs. So the location of the citric acid cycle and pyruvate oxidation, that is the matrix of the mitochondria. The matrix of the mitochondria, that's like the cytoplasm of the mitochondria. It's kind of the inner parts of the mitochondria. So what we're doing is we're going to take acetyl-CoA, we're going to continue to oxidize it, continue to remove electrons from it, and that's why we then create 6 NADH and 2 FADH2s. Okay? More of these electrons are going to get shuttled to these electron carriers that then go to the electron transport chain. And in the process, we end up making 2 ATP. Okay? So for the citric acid cycle, what you need to memorize is that you make 2 ATP. I'll do a different color. 2 ATP. And it's not 2 ATP, 2 FADH2s. Why do I keep using black? 2, 2, 4, 6 are the numbers you gotta know. So 2 ATP, 2 FADH2s, and 4 CO2s. The process of oxidizing this molecule, we're gonna release 4 more CO2s. 4 CO2s, and then you end up creating 6 NADHs. Okay, so 2, 2, 4, 6. So the 2, 2, 2, 2, 2, 2 for glycolysis, 2, 2, 4, 6 for citric acid cycle. Okay, 2 ATP, 2 FADH2, 4 CO2, 6 NADH. But don't forget about pyruvate oxidation. You also in pyruvate oxidation made 2 CO2 and 2 more NADH. That's separate from this. Okay? So sometimes people will combine pyruvate oxidation and the citric acid cycle together, and you would see that you would have 8 NADH and 6 CO2 because you had 2 of those there. Okay, so then we're taking our electron carriers and we're going to send them further along in the mitochondria, and we're going to do something called oxidative phosphorylation. There's 2 steps to oxidative phosphorylation. The first is the electron transport chain. That's where you're going to remove the electrons stored on those electron carriers, and you're going to use the energy that you release from those electron carriers to do a process called chemiosmosis. The chemi is referring to those hydrogen ions, and osmosis is those hydrogen ions wanting to go from more to less, and in the process you make most of your ATP. And I'll show you those reactions in a second. But that's like, if you understand this slide, like you're in a pretty good spot for cellular respiration. The electron transport chain is the inner mitochondrial membrane. That's the location of it. Location of chemiosmosis is a little complicated. I'll show you that when I go over oxidative phosphorylation. Okay, so moving on. This slide is just showing you glycolysis. So just showing you the energy investment phase. So those are the first 5 reactions where we have to invest 2 ATP. And then the energy payoff phase, that's reaction 6 through 10. That's where you get out 4 ATP. But again, because you had to invest 2 ATP and you only made 4, you net 2. So notice the 2s. 2 pyruvate, 2 waters, 2 ATP, 2 ADHs. And technically you get out 2 H pluses that can be used for that hydrogen ion gradient and the oxidative phosphorylation. So 2, 2, 2, 2, 2. Okay. A little bit more on the citric acid cycle. As I said, you don't have to memorize like the actual reactions of it, but keeping track of the carbons is important, meaning you want to understand that pyruvate is a 3-carbon sugar. It comes into the mitochondria by active transport. Now remember, there's 2 molecules of pyruvate. We had to do times 2 because glucose was a 6-carbon sugar. That's why you have 2 pyruvates. This will come in. Pyruvate oxidation is that step before the citric acid cycle where we're going to make that 2-carbon acetyl-CoA sugar. And we make that because we lost a CO2. So we make 2 CO2s here and 2 NADHs because we have to do this cycle times 2. Okay? Then we send that acetyl-CoA through the cycle. And in the process, you're going to lose, this is times 2. So you get 4 CO2s here. And then this is times 2. So in this portion of the cycle, you get 4 NADHs. But you also make 2 more NADHs here. So that's why you get a total of 6 NADHs total. Remember, 2, 2, 4, 6. So you get out 2 ATP, 2 FADH2s. That's what you see right here because you do that times 2. 4 CO2s. So you had 2 CO2s here, and I'm sorry, not that CO2. The 4 CO2s comes from this step right here. So 4 CO2s, and then 6 NADHs. So 4 of those NADHs come here, and 2 come from here. Okay? Yeah, Serena? Are you asking about this CO2 right here? That is technically separate from the citric acid cycle. That's CO2 loss there. That's part of pyruvate oxidation. So like they separate pyruvate oxidation from the citric acid cycle. Sometimes, like I'm pretty sure when I did AP Bio, they combined it all as one. So like if they were to combine, if they were to say how many carbon dioxides are lost in the pyruvate oxidation and the citric acid cycle, you would have to do 4 CO2s plus 2 CO2s, so it'd be 6 CO2s. I know it might seem a little confusing, but that's kind of what's going on there. And remember, this is the matrix of the mitochondria. Okay, we good on this? No? All right, so then getting into the electron transport chain. So going over the oxidative phosphorylation process. The purpose of all of that mess was to make those electron carriers, the NADH primarily and your FADH2, and you see them right here, NADH, FADH2. So this represents stored energy. So we're gonna do a set of oxidation reduction reactions where we pass the electrons from the NADH to the different proteins in the electron transport chain. So this molecule is going to get reduced because it gains these electrons, but then it's going to pass these electrons to the next molecule in the electron transport chain. So it then gets oxidized. This gets reduced, but then it gets oxidized by Q. Q gets oxidized by 3. 3 gets reduced, but then it gets oxidized by 4. You don't have to memorize the order or the names of these. Just know the electrons get passed from protein complex to protein complex to protein complex. And there's energy that gets released from the passing of those electrons. That energy is used to pump these hydrogen ions from the mitochondrial matrix. This is the matrix of the mitochondria. That will give the matrix of the mitochondria a low concentration of hydrogen ions because they're all moving out here. This is the intermembrane space. So it's important to remember that the mitochondria is double membraned. So you have a membrane here, but there's another membrane here. So in between those two membranes is the intermembrane space. So the intermembrane space, because we pumped all those hydrogen ions out there, it's now going to have a high concentration of H pluses. Okay? Now, we talked about diffusion. The basic idea of diffusion is things want to go from more to less. These hydrogen ions want to go from where there's more of them back into the matrix of the mitochondria where there's less of them. The only way they can get back into the matrix of the mitochondria is through ATP synthase. And so, I told you ATP synthase, that's what this is right here. This is the world's smallest turbine. Think of the hydrogen ions as like wind, like a wind turbine. The energy of these hydrogen ions flowing through the ATP synthase will take your ADP plus a phosphate and will turn it into ATP. And ATP, that's our goal. That's what we wanted. That process of those hydrogen ions moving through ATP synthase, that is what's called chemiosmosis. The chemi is referring to the chemical H plus. Osmosis, this process of things going from higher concentration to lower concentration. Okay? And when you combine electron transport chain and chemiosmosis, this whole process is oxidative phosphorylation. So make sure you keep those three terms straight, because it's easy to kind of lump them all together. One more thing that I didn't mention. The final electron acceptor in the electron transport chain is oxygen. I mentioned that on like the very first slide I went over. That's why we have to breathe in oxygen, because oxygen is really, really electronegative. It's the final thing to accept those electrons, and when it accepts those electrons, it then creates water, H2O. Okay? I think that's everything here. Is there any questions on that process? A couple more things before we take a break. This slide I already showed you. I won't do the whole speech again, but as I said, this is like the main summary slide for cellular respiration. Pyruvate is a very key juncture molecule. What I mean by that is we took glucose, we turned it into pyruvate. Now, if oxygen is present, we can do cellular respiration, right? Because oxygen was that final electron acceptor in the electron transport chain. If we don't have oxygen present, we don't bother doing pyruvate oxidation and the citric acid cycle. Now, if we don't have oxygen present, then we still need to make ATP. So say you're working out really hard and you run out of oxygen in your cells don't just like die, they will do something called fermentation, where we will still make ATP. Because remember, when you did glycolysis, you made 2 ATP. So we can still keep making that 2 ATP from glycolysis in the absence of oxygen. That's where we do either alcohol fermentation or lactic acid fermentation. So I want to go over both of those. So alcohol fermentation, this is how alcohol is made. You're like, Mr. Blackford, how is beer made? Well, let's talk about that, right? Like here you have glucose. That glucose does glycolysis to make pyruvate. And in the process, you make 2 ATP. It's not a lot of ATP, but it's better than nothing. But there's a problem. Even if you keep adding glucose to the cell, the issue is in order to make that ATP, you have to have NAD+, because that reaction of taking glucose and turning it to pyruvate, you use NAD+ and make NADH. So we have to regenerate the NAD+, right? We got to find some way to get more NAD+. This is mainly like bacteria cells that do this. How they do that is they take pyruvate, they turn pyruvate to something called acetaldehyde. You don't have to memorize these structures, but know the names. Know that pyruvate goes to acetaldehyde, and in the process, acetaldehyde is a 2-carbon sugar, pyruvate's a 3-carbon sugar, we lose a carbon dioxide. So you just ripped off that CO2. Then what we're going to do is we're then going to reduce acetaldehyde and we oxidize NADH. Meaning we're taking the electrons stored in NADH and we're going to add those electrons to acetaldehyde, making ethanol. Another way of saying that is we take the hydrogen that's on NADH and we're going to attach that hydrogen to that carbon to oxygen bond, creating, or not carbon to oxygen, but attach it to this oxygen, creating OH, an alcohol group. That's why they call it ethanol, which is an alcohol. Okay? And when you lose that H, it becomes NAD+. And the purpose of all of that is now we have the NAD+ we need to do more glycolysis so we can make more ATP. Okay? So for alcohol fermentation, no, you go pyruvate to acetaldehyde back to ethanol. Or just to ethanol, not back to it. Okay. Now, what our cells do is something called lactic acid fermentation. And I think a lot of you have heard that word lactic acid when it comes to like working out and stuff. And that's what happens, right? If we don't have enough oxygen because like maybe you're out of shape or something, you know, your body still needs to make ATP to run its cells, but to make that ATP, you have to regenerate the NAD+, just like we saw in alcohol fermentation. But the difference between alcohol fermentation and lactic acid fermentation is we don't have acetaldehyde. We go straight from pyruvate to, it's a different molecule, but a similar reaction. We go straight from pyruvate to something called lactate. If you have a hydrogen here, that's called lactic acid. Okay? Anyways, we don't remove a carbon dioxide here like we do in alcohol fermentation. Oh yeah, by the way, the like beers, like carbonation, that comes from like the carbon dioxide lost here in that reaction. If you're curious. Anyways, it's the same type of reaction. We are going to oxidize pyruvate, or I'm sorry, we're going to reduce pyruvate, and oxidize NADH. What I mean by that is we take the hydrogen on NADH and we're going to attach it to that carbon to oxygen bond, creating this alcohol group there, that O to H bond there. Okay? And in the process, you regenerate your NAD+ so that you can keep doing glycolysis and making that 2-ATP. Okay? All right, last slide before we take a break. You may be wondering, well, all right, Mr. Blackford, you say glucose is what we use in cellular respiration, but like I don't just eat straight up glucose for all my meals, right? Like I eat like proteins and fats, and like how do I get energy out of them? Well, kind of the same way. Proteins and fats, they all at some point feed into the cellular respiration process, but they feed in at different points. You don't have to memorize where proteins feed in, because they feed in at many different points, or where fats feed in. Just understand that idea, that fats and proteins will feed into the cellular respiration process at different junctures. Okay? It's really all of this when we went over like unit 1, I said you got to start thinking of things like these Lego blocks where you can rearrange them and you can make a fat become a protein. You just got to rearrange the atoms, maybe add some different atoms, but you can rearrange these molecules. Okay, we'll take a little break.